Hello, and welcome to the DNA 2.0 webinar on Mammalian Expression Vectors and Panels. I'm Laura Whitman, Director of Biotech Marketing for DNA 2.0, and I'll be your facilitator for this webinar. Before marketing, I was a research biochemist in both academic and pharmaceutical labs. We'll start in just a minute, but first a few housekeeping items. Feel free to ask questions throughout the webinar. We'll do our best to answer when possible or at the end of the webinar. You can submit your questions by typing them into the question box on the right side of your screen. If your question is not answered during the webinar, we will email you an answer within the next 24 hours. We are recording this webinar, and it will be available for viewing on our website in about 24 hours. You should receive an email with the link. I'd like to thank you for joining us and hundreds of researchers from around the world to listen to this webinar. At this time, I'd like to introduce our speaker, Kate Caves. Kate is the DNA 2.0 Mammalian Laboratory Products and Services Manager. Kate has worked in molecular biology research for many years with a focus on protein expression in mammalian systems. Thanks, Kate.
So thank you everyone again for joining us today. So today we're going to be talking about our mammalian expression vectors and panels. So here's a quick agenda for what we'll cover today. I'll give you a brief background on our company and our capabilities. We'll discuss our new service, Vector GPS, and the catalog products that complement the service. We'll discuss the technology that helps make this possible, the Electra vector system, and then we'll talk about how to get started on your project. So a bit about our company, for those who are not familiar. We are based in Menlo Park, California, and have been around for just over 11 years. We are privately held and wholly employee-owned, and what we do is leverage our expertise in gene synthesis and bioinformatics to provide bioengineering solutions for our customers. So there are two basic ways we like to help customers along the path of product development. One is by providing tools such as gene construction software, such as Gene Designer, which I'm sure you are familiar with, synthetic genes, and vectors. We also try to provide engineering technologies that help determine the relationships between sequence and function. And in the simplest form, gene synthesis is just providing access. At least that's how we see it. And it allows you to obtain known genes and allows tailoring to facilitate the use. But in the fullest sense, we found that we can use gene synthesis to allow us to help take complete control over the information in genes. And really the information that's relevant to protein expression, and as well as what's relevant to the translated protein sequence. And the key, obviously, is knowing how to control that information. So what we try to do is we offer engineering services that leverage the potential of gene synthesis to help our customers search for these solutions in a sequence function space. Today we'll talk about the vector level. So ultimately our interest is not simply in just genes or proteins, but the entire system. Whether it's a production system for a protein therapeutic or an engineered algae producing a biofuel. Much of these systems can be addressed at the sequence phase. Gene synthesis technologies and other developments in synthetic biology have given us powerful means to manipulate DNA. And as a gene synthesis company, we're really invested in how to explore the relevant sequence space for various applications. So today, like I discussed before, and why you're here today, is to look at the vector space. So we are approaching our vector optimization by experimentally interrogating the system. And this is very similar to our approach for codon optimization, which I'm sure many of you are familiar with. And really, our method is to create a diverse set of vectors and then test their expression in a heterologous host. And then we let the data tell us what's important. And a key feature of this approach is that we're using design of experiment methods. And this really allows us to broadly vary the vector design variables while minimizing covariation between these variables to most effectively explore the space. So combined with our gene synthesis capabilities, we're able to readily and rapidly sample any variable over any range we can cover. So we start with an in silico design, the design of experiment, and we create infologs. And these infologs, in this case, are individual vectors. We've synthesized hundreds and hundreds of vectors for the purpose of our vector GPS services and products. And we're testing in a variety of host systems, including mammalian host systems, E. coli, and yeast. We'll touch over all of them today. And from these, we're able to build these predictive algorithms. And our work is, for our codon optimization, was published in PLOS One. And we're currently in the works of publishing for our vector GPS services. So this is just an overview of what it looks like specifically in the context of the vector. So specifically, for the case of optimizing vectors, we create an initial set of vectors with a diverse combination of elements. And we started our internal vector GPS work in mammalian host systems with the goal of exploring the modularity of components so we can optimize the expression for our customers interested in increasing antibody expression. But now we've expanded into a much larger space than that. And we see ourselves in an optimal position as a company that performs gene synthesis, as our element portfolio is quite large, as you can imagine. But as you can imagine, just looking at this picture, the full factorial combinations to explore is very large. So applying our design of experiment principles can allow us to sample the space more effectively without creating bias. So we started our experiments looking at the elements that you see here. So we tried a variety of different enhancers, promoters. We got down to the intron level. For bicystronic expression, we've looked at irises in 2A. We've looked at different localization signals and polydenylation signals. And we used our in-house and IP-free, intellectual property-free GFP dasher for the initial experiments. And I'm sure many of you are interested in this as well. What elements did we start with exploring? So as you can see here, there's an extensive list for the initial set of experiments that we explored. A lot of them are classic. You've probably seen them. And we've gone beyond, based on our round one initial data, to design and re-engineer synthetic versions of many of these elements. So the graph here shows our initial findings, and we've also built in additional vectors that we found based on our round one data. Here I show you the performance of the 24 vectors that have been tested in HEK 293 cells and CHO cells. And as I mentioned before, we used Dasher GFP, and so we measured total fluorescence of transient expression, and it was measured at 72 hours. As you can see here, HEK and CHO cells are showing distinct preferences. And we are able to identify one combination of elements that express well in both cell lines. However, as you can see, vectors that perform very well in CHO perform poorly in HEK, and vectors that perform fairly well in HEK were less than optimal in CHO. So we're currently working to explore more of this sequence space, but the take-home message for this slide really is that HEK and CHO do have distinct preferences for vector element combinations. I also have marked here 3 vectors of importance. So PJ609 and pcDNA 3.4 have typically been used as the workhorses and industry standard for HEK and CHO expression. As you can see here, through design of experiment in our projects, we've been able to identify a large number of vectors that express at higher levels. Here, a lot of these vectors are available as catalog items. Here's one vector that performs very well in HEK and CHO transient that's also available. And furthermore, the data here shows that we're able to successfully create a predictive model for HEK and CHO. What the graphs are depicting here is measured expression versus predicted expression. So on the x-axis you have measured expression, and on the y you have predicted expression. Blue represents the model points, and red represents cross-validation points. And as you can see, the model's fairly tight. And if you try to overlap you can see there is very little overlap, again showing that HEC and CHO do have distinct preferences. So we could do this with a variety of cell lines in transient and stable, and I will go on to show you more data on that. But the cool thing is that we can predict the expression, we can predict expression of specific vector element combinations in a system. So this is my favorite data, and I think it's really exciting. So we're able to use multiple regression analysis to weigh the contribution of each element to expression and HEK and CHO. So each red and blue pair represents a different element, and you can see here we have enhancer promoter combinations, polydenylation signals, and viral amplifiers. Blue represents the contribution of expression in CHO, and red represents the contribution to expression in HEK. From this data, as you can see, we're able to identify specific element preferences for each cell line. And we're also able to identify elements that hurt expression, and many of which are commonly used in current classic industry standard HEK and CHO vectors. You can also see here on the right that HEK and CHO have strong preferences for viral amplifiers. And in this test, we looked at ORIP, SV40T, and eBNA. So this is really interesting data that surprises a lot of our customers. But furthermore, this data specifically allows us to identify elements of focus which we can re-engineer. So this is what we're focusing a lot of our time and resources on right now. And when you think about just breaking apart and focusing on which, for example, promoters contributed most to expression and how we can engineer them to further enhance expression, you can really display the power of design of experiments. So we are able to deconvolute the pieces of each vector, model it, and then perform more rounds of experimentation on each system and on the specific elements. So from this, we've been able to re-engineer vectors and create far superior vectors. So we also started doing this in mammalian HEK and CHO systems for stable expression. So these, again, we use our in-house IP-free Dasher GFP, and we measured total fluorescence again. And this is 2 weeks post-transfection, and they were selected using pyramycin and passaged 2 times. So we saw fairly stable integration and stable expression of the gene. And as you can see here, again, looking at these 2 industry standards, PJ609 and pcDNA 3.4, we could do a lot better for stable expression. And HEK and CHO, again, are showing distinct preferences for vector combinations for stable expression. And this data, these vectors really are powerful tools for stable expression. For these top vectors that are available as catalog items, we see that over 80% of the cells we select are GFP positive and exhibit stable expression over many passages. And we've sent a lot of these top expressor vectors for third-party validation. And one of our customers at a reputable pharmaceutical company was so impressed with these vectors, and he reached out to just ensure that the data he is seeing and what he was seeing and throughout selection mirrored what we were seeing. The stable integration and stable expression really is quite remarkable. So we've added a higher performing transient stable vectors to our catalog, and here's just a brief overview of the vectors and features that are available as catalog items. So they're broken down by numbers, so we have groups of PD 600s and 2600s as transient vectors, PD 2500s as stables, and PD 1100s as lentiviral vectors. So they're available for bicistronic expression with IRAs and 2A elements. We also have expression monitoring using fluorescent proteins such as our IP-free Cayenne RFP and our IP-free Dasher GFP. We also have added additional elements for localization monitoring such as N and C terminal fusions and fluorescent proteins, the same IP-free GFP and RFPs. And the stable vectors and the lentiviral vectors are available with antibiotic selection markers. And at the end of the webinar, I'll walk you through where to find these on the website and how to order them using a vector selector tool that we've made as interactive specifically for this purpose. So we've also performed vector GPS in E. coli. So we use different promoters, RBSs, secretion signals, and OREs. And what we found, and I'm sure many of you have seen this in your own work too, is that expression levels are influenced by the promoter, the ribosome binding sites, and the plasmid copy numbers. And the total expression level does not necessarily correlate with the level of soluble, active, or secreted protein. But proteins can be fused to affinity type secretion signals and different solubility enhancing domains or fluorescent proteins. So we try to combine, we try to create as many optimal combinations of these elements to best express a wide variety of proteins. But for secretion, we generally recommend the rhamnose promoter because it's titratable and at inducer concentrations and can be selected so that most or all the protein synthesized is secreted. And we have also found, and I'm sure you've seen this, that very strong promoters tend to overwhelm the secretion machinery and can lead to large amounts of unprocessed or insoluble protein in the cytoplasm. So different proteins are showing strong preferences for different secretion signals, and I'll show you this over 2 case studies. So basically what we've done is we've created a panel of 14 vectors because we recommend testing in panel format to include in your experiments various combinations of these elements. And this panel is available as catalog items. And this has become a rapidly growing and popular catalog item of ours because customers have chosen just to store this in their freezer. And it comes in Electra cloning reaction format. So whenever you have a new gene you're working on or that difficult to express gene, you can pull this out, and in a 5-minute benchtop reaction, you should be able to identify quite rapidly a vector or multiple vectors that enable you to efficiently express your gene of interest. So here's the first case study. So we tested our IP-free Dasher GFP in a selection of vectors from the panel. And if you see below, you can see the different elements that are incorporated in each vector. So the top graph is an overall look at fluorescence, total fluorescence units measured, and then the bottom one below is a zoomed-in version, so you can compare, so you can better see the differences of the lower expressing vectors. And you can see that there's a wide range of expression. And furthermore, you can see that different leaders work for different secretion mechanisms. So our panel includes a wide variety of selection of leaders, and you can see that this greatly affects secretion, and you can see we get a wide range of secretion. And here's just another quick case study and where we looked at cutinase. And here I'm showing a subset of the data from the expression panel. And what we did was cultures were induced by addition of rhamnose in 3 concentrations, and we ran uninduced cultures as the negative control, as you can see at the end of the gel. And as you can see in the gel, there are clear differences in expression and processing of the cutinase protein with different secretion signals in the periplasmic fractions, which is shown by the green arrows. So the highest expression levels of cutinase in periplasmic fractions that were observed had the secretion signal OMC and MAL-E. We also saw strong levels of cutinase expression with DSBA and TORT-T. However, as you saw with the GFP of Dasher, the secretion is affected by the secretion leader. So we really highly encourage you to explore a variety of different secretion leaders. So I'm quickly going to run through yeast as well. So this is the last host we'll discuss today. And we've designed a series of vectors for expression in Pythia and Saccharomyces. And we've, again, made a panel of various secretion signals as we encourage you to test these in panels. And our Pythia vectors have been designed for cytoplasmic or secreted expression. We have different promoter choices for inducible or constitutive protein expression. And we also have a choice of signal sequences available for secreted expression. And for our Saccharomyces, you can see here mapped out as well that we have different promoter choices We have GAL1 for galactose-inducible expression. We also have TEF, ADH, and GPD for constitutive protein expression. And these are available with a wide range of oxytropic selectable marker choices. We have LU2, URA3, TRP1, and HIS3. And I'm going to show you really quick data. Here's another panel case study using cutinase with various secretion signals. So we measured the cutinase levels using SDS page. And as you can see here, the secretion signal does make a difference. So on top, I'm showing you picky, and on the bottom, you can see Saccharomyces. So again, these are all available in a panel format, and we highly encourage you to try and to explore the space in more than one vector. So the thought of testing these vectors probably seems daunting and like a lot of work. So the way we've been able to do it in-house is with the Electra vector system. So we use Electra cloning to perform all the experiments I've discussed here today. And it's really allowed us to do that in a rapid and cost-effective manner. So for those of you who aren't familiar with Electra, it truly is an enabling technology. It's a 5-minute rapid benchtop reaction. And we also have our gene designer software that can help you design your gene to make it compatible with the Electra vector system and does all the design work for you as well. But as always, we can do the cloning work for you. That is not a problem. So I'll just briefly go over the Electra vector system. So it's based on a type 2S restriction enzyme SAP1. And SAP1 recognizes a 7-base pair non-palindromic sequence and cuts just outside to the 3-base pair overhangs. So we utilize that first overhang as an ATG. And at the end, you can either have a stop codon, or you can have some kind of small amino acid, like a glycine, for a terminal fusion if you would like. And what you can do is, all of our vectors come as daughter vectors with complementary overhangs to the ATG or to the stop codon or whatever you choose, whatever is at the end of that, whatever is at the end of your gene. And when you mix your mother and daughter, it's a 5-minute benchtop reaction, and you transform and plate, and 95% of the transformers will have your orth of interest. That's what we've found. If you order your gene, it can come in the P-mother format with these overhangs to make this easy for you to test. But if you have your gene in another vector and you'd like to use it, you can also just use primers to amplify out your gene with these complementary overhangs and just mix your PCR reaction with your daughter expression vector. There's no license on this and no restrictions. So it's a fast, easy, efficient system. You don't have to worry about intellectual property, and you can make your system compatible with ours. And it's really enabling to do this fast and easy. So how do you get started exploring this space? I'm sure this is a new space for a lot of you, and that's why you're here. So if you already know what you want because you're very familiar with your host and you're working in a well-defined host, and you're just, you know you want this specific combination, you can always just try one of our single expression vectors. They're readily available on the website. We have over 400 vectors, and I'll show you how you can use our interactive tool to easily select the ultimate combination for your specific project. We also have these expression panels that I've been discussing. So we have them for mammalian, E. coli, and yeast systems that you can order. And they have 14 vectors, and the subset of vectors has been chosen to help improve expression over a wide range of protein types. And lastly, if you want the very best vector for your system, you could do vector GPS, which is the experimental process that we've done to gather the data that you've seen here today. And vector GPS truly is a powerful tool to help you optimize expression and find the very best vector for expression, as you can see in the data that I've showed you here today. So I'd like to share with you a quick vector GPS case study. We've been very successful using vector GPS to help one of our biofuel customers. So they're working in a unique bug with a proprietary genome and with a unique protein that was particularly difficult to express. They came to us with very little knowledge on expression for their system, and they had very little knowledge on how to improve expression. So using our gene GPS codon optimization technology and vector GPS, we're able to help develop them a proprietary platform that's completely licensed and IP-free, as all of this is done as a fee-for-service project. And furthermore, the turnaround time was rapid. Because of the power of DOE, they tested 96 vectors and were able to find something that worked very well for their system to increase their expression by 8-fold. So for those of you working in unique hosts and unique proteins with little knowledge and not sure which direction to go, or if you've already tried optimizing your gene and you still can't get it to work, this is definitely a space to explore. So now I'm just going to demonstrate to you the website and how to order. So we'll start at our home page. So we're located at www.dna20.com, and the web page is loading right now. So when you get to our web page, you'll see, I'm sorry for the lag. So I think the easiest way to get here, and the best way to get there is if you just go to the top, there's a products tab, and under expression vectors, you can click on your host system. So specifically, because this is a mammalian focused talk, we'll go to our mammalian expression vector page. So on the first page, you'll see an overview of basically what I've communicated with you today, which is the vectors that we have available and the different combination of elements and types of expression vectors that we have. If you click on the expression tab, you can find this really handy vector selector tool. So this is an interactive chart. So as you drag over, so here's the vectors. This is the same exact data graph that I showed you in the slide. So we're, again, looking at each vector, and we're looking at how it expresses in HEK and CHO. So we have this for transient, and then below we have it for stable expression. You can just hover over one of the vectors, and you can click to add it to the cart. So if you're working in transient HEK and you want something that works very well for you, you'd most likely probably select this vector. You can click add to cart right here. And then you can also order the control that will come with a Dasher GFP to use alongside as well. And you can, if you just click here, and then as you order more, you get a larger discount. We're really trying to change the mentality of our customers and encourage them to explore into this space. And in order to really see the power of trying these different elements, you need to try more vectors, as it is protein dependent. So this is an easy way to order to cart. After you've ordered to cart, it will turn green, the vector. So you can go through and you can select as many vectors as you like. If you have a specific desire and you need help selecting, you can always contact us. We are always on the other line with PhD level technical support. We can help you select vectors for your specific project. And these are, however, this is the actual data that we've gathered in-house in our lab, so this should guide you efficiently. Furthermore, we're always adding new vectors to this graph, because as we interrogate the host and do this iteratively in rounds, we are identifying more optimal, better combinations for vectors. So this graph will constantly be updated with better vectors. So you can do this for transient or stable expression. And as you can see, once you order 4 or more, or both panels, you get 30% off. So this significantly reduces the vector costs. And then if you just want to try a panel and you don't want to choose, we've selected 10 vectors that we found work over a large range of different protein classes, and you can order as a full panel and receive 50% off all 10 vectors. So here again, on this page 2, we go through the different ways for you to explore the vector space. And then if you click on the other tabs, we go into more detail about the vector elements and how they're built. And we also provide more information on each feature of the vector. So one of our goals at DNA 2.0 is educating the customer as best as we can. So we do this by trying to provide as much information on our website in an effective, clear manner. So please explore the website to find more information on each feature. So we have different tabs for transient and stable as well. Here's the backbone of our stable vector, and then each vector on this graph is some variation of this backbone. We also have lentiviral vectors, and we currently have a large in-house R&D investigation to improve our lentiviral expression vector. The vectors that are available can be found on this tab. And we are performing vector GPS technologies on the lentiviral vector, so stay tuned. We have exciting data in the works, and hopefully we will have a webinar on that soon for you as well. So you can explore the different expression vectors under this expression vector tab. We have pages on bacterial and yeast as well. And then for those of you interested in vector GPS and really finding the optimal vector for your specific host, you can come here to find more information and then contact us today, and we'll put you right in touch with one of our vector GPS specialists today. And our vector GPS specialist team consists of half bioinformatics members and half scientists. Okay. So just to finish off, at the beginning of the talk, I showed you this picture. So like I said, we are really interested in optimizing the entire DNA sequence space, the protein level, the gene, vector, and strain level. And today, we were only able to discuss optimizing at the vector level. We currently have webinars, so you can click on this link. You'll receive this PowerPoint after the webinar. You can click on this link to find more webinars that have been recorded on optimizing at the other levels. We also have an exciting protein GPS webinar coming up on September 24th. I encourage you to attend that one as well to explore how we are using machine learning to optimize at the amino acid level. And then for strain engineering, we also have new CRISPR products, CRISPR-Cas9 products that we are launching for yeast and E. coli as well, so stay tuned for that. So thank you very much for your attention and for joining us today. If you have, we'll take questions now, but if you think of a question later, as always, do not hesitate to contact us at info@DNA20, or you can visit our website. We now have a full-time live chat available during normal business hours, and you can use that at all times too to get in touch with our PhD level technical support. So thank you very much, and now we'll take a couple questions.
Thank you so much, Kate. Thanks to everybody for joining us today. We hope you've discovered a valuable new set of tools with which to move your research projects forward quickly and efficiently, and we look forward to working with you on your next projects. We'll now answer as many of the questions that remain as possible, and as Kate mentioned, of course, if you have additional questions, you can always reach out to our entire team by phone, email, or live chat. So, Kate, one of the questions that came up more than once was when looking at the mammalian data, why is there such a big difference between the RFUs when analyzing CHO versus HEC. You note on the graph that the X and Y axis have fairly different numbers.
Yes, so as we know, for transient expression, HEK is better for transient expression. So just in initial first and second rounds, we were able to optimize expression at much higher levels. CHO is a bit more finicky and harder to work with for transient expression. So that's why you're seeing lower numbers. However, we are exploring this space, and we currently have a vector GPS project in the works via a collaboration to explore CHO transient expression to further increase our expression yields in CHO cells. So we're trying to get that number up as best and fast as we can. And we're also working, you're probably wondering, for stable as well. So CHO, HEK still it expresses very well 2 weeks post-transfection. However, CHO is more popular for stable expression and more commonly used, especially in antibody production. So this is the initial data. We haven't built a predictive model yet for CHO stable expression. However, that's one of our other large projects that we're currently working on, and we should have that data out soon.
Thank you. Another question is regarding which fluorescent proteins we recommend. They're curious if we recommend anything besides Dasher, particularly looking for proteins with strong fluorescence, quick maturation, and various colors.
Okay, so I have this exciting slide. I'm sorry that it says Pichia. These are for Pichia, Saccharomyces, E. coli, and mammalian systems. So we have a variety of chromogenic and fluorescent proteins. So the chromogenic proteins are only for E. coli. They're visible by the naked eye, and they're very strong. They do have different maturation rates, which you can find on our website. These are very useful for screening in E. coli. For fluorescent proteins, we have these available in mammalian yeast and E. coli, and we've done quite a bit of characterization work on our RFPs and GFPs. However, we do have quantum efficiencies for all the colors available, and they're very high compared to other industry standard fluorescent proteins. They're bright, they're very stable. Sometimes that's a good thing or a bad thing depending on what you're trying to do. But they've been extensively characterized, so I highly encourage you to visit under the products tab on the website where I showed you the chromogenic and fluorescent protein tab, and you can find which colors are available for which host system you're working in and more characterization data on each.
Great, thank you, Kate. The next question is regarding inducibility, and they're curious, do we have any inducible mammalian vectors for either transient or stable expression?
That's a great question, and that's something that we're spending a lot of time working on here. So at DNA 2.0, as I'm sure you've seen throughout the entire presentation, I've constantly said IP-free. So we're very focused on providing intellectual property-free items for our customers, as the majority of our customers are in industries and biotech space. Right now, there's a lot of IP around inducible promoters, so we're trying to develop our own version of a tightly regulated inducible promoter. It's in the works, but however, I do not see those products coming up in this year, probably early next year. So we currently do not have any inducible promoters, but as always, we can design or build and work in an inducible element into any of the vector formats that you see here. So just contact us, and we can work with you to build your ultimate inducible, stable, or transient vector. But we don't sell them right now because they're not IP-free.
Great, thank you. A good follow-up question just came in regarding IP restrictions. And they note that stable expression vectors in mammalian systems have the WPRE, and that, of course, has IP restrictions. So are there any vectors that don't have the WPRE?
That is a great question. So this is really exciting. since I started at DNA 2.0, we've been trying to develop an IP-free version of WPRE, and we have been able to do that. So some of you may have seen this on our website. So we found HPRE it's an IP-free version of WPRE, and furthermore we find that it's more efficient at enhancing stable expression than WPRE. So if you're looking for a stable expression that is completely IP-free, and it happens to be our best expressor, which is just awesome, this is PD 2509. So write that number down. It's definitely my favorite vector for stable expression. We use it all the time in HAS to create stable cell lines in under 2 weeks. And I've just been getting rave reviews from customers on this vector. It's truly a great find from Vector GPS.
Excellent, thanks. Another question that many people asked was, how many vectors should I use? Can I start with more than one but less than a whole panel? Do I have to order the whole panel? What do you recommend?
So going back to that getting started slide at the very end, it really depends on your project and the resources that you have. Some people have larger screening capabilities, some people don't. So it's definitely when you start, you want to start with more than one vector to even begin to explore the space and to explore the power of manipulating the vector. But it's all going to depend on your resources and your capabilities. As I discussed with you, we have these are all Electra vector formatted. So we try to make it as easy and as quickly as possible to screen. But I do realize that it is difficult to screen large numbers of vectors, especially when you get above 10 or 14. So we've created these panels with the hopes of trying to provide a solution for people who don't have high throughput screening capabilities. So if you can explore the panel, I highly encourage you. Not only is it inexpensive, especially when you look at the price it comes down to per vector, but the elements and the combinations that we cover, it's such a large range. You'll be very happy with your results if you can explore the expression panel.
Great. Another similar question is, what do you suggest trying if the researcher is using another system besides HEK or CHO? Is there a panel or a set of vectors that you recommend for that?
So this is a great question. So if you're working in a host system that you don't see discussed on our website, you can always contact us. We have expertise in a wide range of different host systems. If it's a mammalian host system, what we can try to do, we'll work with you to identify if you do know which elements work well for your system, and then hopefully we can try to create a panel of vectors that have already been assembled to lower the cost and to do this as quickly as possible to start with and gather the initial data from there to design something optimal. But if we don't have something, if we have to, then we'll do a vector GPS type project, and we can do a vector GPS type project in as small as 12 vectors. So we can start there as well. It depends on the resources you have available for screening time and financially as well.
Great. Thank you so much, Kate. At this point, we're going to bring things to an end. It's been a little while. I'd like to thank everyone again for joining us today. We again hope you've discovered a valuable new set of tools with which to move your research forward quickly and efficiently, and we look forward to working with you on your next project. If you have any additional questions, you can always contact us by phone, email, or chat as noted on the last slide. And as a reminder, a copy of the webinar will be available on our website and YouTube in about 24 hours, and we will be emailing you out a copy of the slides. For more information on any of the products and services you've seen today, you can also always visit our website. Thank you again so much, Kate, and thank you so much to our audience for listening.